Welcome back, AP Bio, to Chapter 10.3 as we really dive into the meat of meiosis this time. So, meiosis, you're going to see, is very similar to mitosis. We're going to have a replication event where our chromatin is duplicated. So we have those duplicated X-shaped chromosomes. We go through prophase, metaphase, anaphase, telophase, just like we did in mitosis. But there's a big difference we're going to talk about here. And then also you'll see we now have prophase 2, metaphase 2, anaphase 2, telophase 2. We've got a whole second event of divisions. So we have one replication event, two divisions, and we end up with four daughter cells that, unlike mitosis, are going to be genetically unique, each one genetically different, and each one haploid, or having half the amount of chromosomes necessary. So when we look at this, you can see we have PMAT1 and PMAT2, or those prophase, metaphase, anaphase, telophase. And so that's how we're going to remember them, is we go through the same steps in each division. So we call those the first stages and the second stages, or meiosis 1 and meiosis 2, to denote the two different divisions. You'll notice we only have one replication event, and that's important because if we replicate, we're doubling the amount of DNA we have. If we divide that in half, we're back to the original amount of DNA, and if we divide that in half again, we'll be at half the amount of DNA. So that's going to bring up a good question. At what point in this process do our cells really become haploid? Is it after that first division? Is it after that second division? Well, logic would say if we double half and half, we would become haploid at the second division. But that's not actually the case. We are going to become haploid after the first division. We're going to end up with 23 chromosomes and 23 chromosomes again. So what's going on here, and how does this happen? Well, it's all going to rely in that first meiosis, metaphase 1. So in mitosis, if you'll remember, when they meet in the middle at metaphase, those chromosomes, they line up in single file line so that those replicated chromatids can separate, and each cell will end up with exactly half and copies of every single chromosome. In meiosis 1, we're going to line up in double file line. So they're going to find their homologue, those homologous chromosomes. Remember, each chromosome we have a pair of. We have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, all the way down. And they're going to line up matched up together. And the entire replicated chromosomes are going to part ways. So that is going to result in us technically only having half the amount. So in humans, 23 total chromosomes. So this is going to be key to us achieving a lot of genetic variation and diversity that we'll talk about in 10.4. So it's important to count chromosomes. Remember, a single unreplicated chromosome, one single bar, is one chromosome. When we replicate that chromosome and we have that X shape that are joined together at that centromere, those two sister chromatids, that is still one chromosome. You may be like, wait, what? They're both one chromosome? Yes, as long as they're attached, they're one chromosome. When this replicated chromosome splits, it now becomes two individual chromosomes. So one single chromosome has the potential to become two separate chromosomes when it splits. And that's going to be really important to this process. So when we count chromosomes, because they're going to line up in double file line, we're not going to end up with every single chromosome after this first split, because those homologues are going to separate. Chromatids are not separating. So we don't get each individual chromosome. We get one or the other of each chromosome pair. So since we're only getting one or the other of each chromosome pair, we're going to technically be halving the amount of chromosomes we have in that first division. So we're going to be 2N, or diploid, to start. And after that first division, we're just going to be at N, or a haploid cell. And then when those chromatids separate in the second division of phase 2, we're still going to be at half the amount, or a haploid cell. So both of those are going to be haploid cells, even though it feels like after one, we've still got double the amount of chromosomes. So, important distinction there to understanding the difference between diploid and haploid. So if we look at this all together, meiosis versus mitosis, the big differences are in metaphase 1, we're in double file line with those homologous chromosomes pairing, whereas in mitosis, they line up side by side. We'll talk about a couple other events. You can see there's some mixing of information happening here. And then there's 2 divisions instead of 1. So we end up with mitosis, 1 division, producing 2 exactly identical daughter cells. Those final cells are going to be diploid. In humans, that means 46 chromosomes or 23 pairs. And it's only going to take place in our somatic cells or most of our body cells. With meiosis, we have 2 rounds of divisions. That's going to give us 4 daughter cells, each one genetically unique. We have haploid cells, so they're going to have 23 chromosomes with no pair, no homologs. And this is only going to take place in our gametes, or our sex cells. All right, that's the end of 10.3. See you next time for 10.4.